Welcome back as we wrap up chapter 15. So we now know that genes are on chromosomes. We know that because of that, we've got some kind of weird inheritance patterns like linkage and sex linkage. So now we're going to kind of talk about how the fact that genes are on chromosomes can kind of allow us to screw things up. And when I say allow us to screw things up, I mean allow for mutations. These aren't always bad. This can allow for us to get new varieties of things. In some cases, it can allow for speciation. But these are things that are still going to happen kind of accidentally and lead to those changes, good or bad. So chromosomal screw-ups, chromosomal oops. And so the most common one that we'll talk about will be nondisjunction. And this is just where the chromosomes fail to separate. So this could be where you have the sister chromatids in meiosis II, and they're supposed to split. That can screw up, and so you end up getting both of the sister chromatids. So essentially you get both of mom's chromosomes, if you will, two copies of mom's chromosome, or you get two copies of dad's, because the sister chromatids both came from the same parent. The other way you can screw this up is in meiosis I, you line up the homologs. So you've got mom's chromosome and her copy, the sister chromatids, dad's chromosome and his copy, the sister chromatids, and you might, for a particular type of chromosome, grab both and just say, I'll take mom's and dad's, instead of just taking mom's or dad's like you're supposed to. And so if that happens, what the end result will be is aneuploidy. Now, aneuploidy is when you're typically going to be 2n plus 1 once fertilization occurs, so the offspring, the zygote, or 2n minus 1. Now, if you're 2n minus 1, that means for a human you'd have 45 chromosomes, and we would say that's the result of monosomy, because you're going to have one of a specific type of chromosome. So normally for chromosome 1 through 22 and for the sex chromosomes, we have two of each. In monosomy, for one of those guys, so for chromosome 3 we could say, you just have one copy. You didn't get one from the ovum and one from the sperm. You only got one from either the egg or the sperm because something screwed up and so it was missing it. The other way you get this is trisomy, where you get a third copy, so that whenever you have monosomy for one gamete, the other gamete gets more. Right? We didn't lose a chromosome. So if you look down here at the picture, you'll see that this screwed up and it was supposed to separate the sister chromatids here, but because it failed to, one of the ovum got two chromosomes instead of one, and the other one had none. And so that means that one of the offspring now is going to have essentially a trisomy. It's going to have where it's going to have an extra, and the other one's going to have fewer. It's going to have a monosomy. So for humans, that means 47 if you have a trisomy, and this is an example would be Downs, where you have 3 copies of chromosome 21. There are monosomies that occur, although they're a little bit rare. You do have Turner syndrome, which is often written XO, because you only have one X. You don't have anything else. So you're typically shorter, slight mental retardation, webbed neck, usually infertile. There's also going to be several trisomies besides Down syndrome with sex chromosomes where you can be XXY, that's Klinefelter's. You're typically male, just with kind of reduced maleness, if you will. You can have what's sometimes called the super female, XXX, who's typically pretty much a normal female. And you can have a super male, which is XYY, which is typically a normal male for the most part. Maybe a little bit more testosterone, maybe a little bit more likely to take risks and end up in, I don't know, prison. But overall, I think that these are pretty normal. You wouldn't look at somebody and go, oh my gosh, that's a super male. No, you're not going to see that they're XYY. So with the sex chromosomes, we do have quite a bit of this that can work and we can survive. Now the other type of screw-up is polyploidy. And this is where you get an entire extra set. So you don't just do this for chromosome 1 that you screw up. You just do this for chromosome 1 through 22 and the sex chromosomes. You're just like, I'll take all of these, thank you. And so one of them is going to have a complete extra set, which makes it 3n. In some cases, this can happen with both of the sex cells, the sperm and the ovum, or it can happen over generations multiple times. So you can get things that are 4n and on. There are things that are decaploid like 10n that exist. Now they're typically plants because they're pretty open about getting extra sets. They're oftentimes healthier, so a lot of our crops like strawberries are actually polyploid. You do get some invertebrates are fine with this. Some fish are fine with this. Some amphibians, specifically salamanders, tend to be okay with this. You don't tend to see this with things like reptiles, birds, or mammals. But it does happen in nature. It's not that odd. Polyploidy, extra sets, it's a screw-up. May be good, but it's still a screw-up. Now the other way we can screw things up is going to revolve a lot around crossing over. When you do crossing over, you can botch it because you're taking chunks of chromosomes and moving them around, detaching them, and so lots of stuff can happen. You can get a deletion, so this will be the first type. In a deletion, you just kind of lost the piece. It's just no longer there, it's gone, and so the chromosome is missing a chunk. So deletion, pretty simple, miss a chunk. You know, you tried to do crossing over where you tried to swap, but the other one didn't reattach, so now you're just down. You're missing information. Not typically very good. Duplication would be like where you have a swap, but you don't give up your chunk, you just take the extra chunk and stick it in there. So it's kind of like you saying, we'll trade, but then you just keep all of it. And so now you have extra. So that would be a duplication. Inversions where you go through and you're like going to swap something, so you like take a chunk of your hand, so to speak, to swap with somebody else, but then you attach it reversed. And that does not work the same way it did before. So inversion, you're swapping it in the process of doing this. Insertion, the idea with an insertion is that you are going to take a chunk of something from another chromosome, typically, and add it to a different chromosome. So I'm taking, in this case, you can see it's a chunk of chromosome 4, and I added it into chromosome 20. So this is where like a deletion would occur for one of them, where it kind of loses its chunk. It didn't reattach what it was supposed to. But that lost chunk could find its way somewhere else, and while that one's going through this process, it slides its way in. So now we have one guy who's kind of down a chunk, and the other guy has this extra chunk, but it's not a duplication. It's not the same chunk. It's a chunk from some other chromosome that it's not supposed to have. And then translocation is what you have when two chromosomes that are different from each other, that should not be doing crossing over together, successfully do crossing over, and by successfully I mean they do swap. So this isn't like insertion where you took somebody else's stuff but you didn't give anything back. This is where you say, hey, me and you will go ahead and switch, but imagine if you're two completely different heights and you try to switch from the knee down. One of you is going to have way too short of a leg from the knee down. The other one has way too large of one because you weren't supposed to switch. You're not equivalent. And so when that occurs, we get a translocation where two chromosomes did crossing over that should not have done crossing over. And that's going to be bad as well. One of the cool things that we have is genomic imprinting, which is kind of an oddball, but it is related to chromosomes. We'll discuss more about how this works because it's kind of more based upon epigenetics, which we haven't discussed yet and won't till chapter 18. But it's this idea that where you get a gene from, like which chromosome it comes on, matters. That if you get one from your dad, so via the sperm, you might get a different result than if it came from the mom, via the ovum. So one example of this is there's a screw-up that you can have on chromosome 15. Now, if that screw-up ultimately occurs on mom's chromosome and is passed to you, then ultimately you can get Angelman's, where you get smile a lot, severe mental retardation, inability to speak. But that only happens if this particular screw-up occurs on chromosome 15 via mom. If the same thing happens, but you get it via dad, at that point you'll get something called Prader-Willis. I believe I pronounced that right, maybe not. But this one basically leads to having like insatiable hunger. And so you might see pictures where you have people that are large, but then they also have what's like chain refrigerators because they just don't feel full. And so they keep eating, so you have to restrict them. There's some other issues they get with it, but that's one of the common ones you'll see. So the same thing, the same screw-up, if you will, has a completely different disorder that it causes based upon which chromosome that screw-up occurs on. So that's this idea of genomic imprinting, that sometimes, not that often, but sometimes at least, it can matter where you get this screw-up from. I believe that's it for this section, so we'll wrap up 15, and I'll see you guys next week, and we'll start on 16 afterwards. Take it easy.